Very small things. So we're going to start lesson 1 with the atom. We got electrons, protons and neutrons. Neutrons and protons are in the middle. We call that the nucleus. And then we got the electrons buzzing around the outside. We've got -1 for electron charge, +1 for a proton, and then neutrons are neutral. Lesson 2 was elements, mixtures and compounds. We've got an element here. You can see that they're made up of one type of atom. So all these are the same type of atom. Compounds, if I just zoom in on this a little, more than one element chemically bonded. So it's 2 elements there chemically bonded. Quite hard to separate these. And then we've got the mixture. This is just a mix of everything. So you can have 2 or more elements or compounds and they're not chemically bonded. Some are, but some are not. And they're really easy to separate. Lesson 3 was electron structure. There's a certain way that we fill these shells. On the first shell, which is this blue one down here, it can only fit up to 2 electrons. But your second and third shells, we can take up to 8 electrons. Now I haven't put 8 electrons in this outer shell. I've only got 4 electrons in this outer shell. I could have had another 4 because they can take up to 8. But I'll tell you why I've done that now. Because when we do our electron configuration, we'll say that there's 2 in the first shell, 8 in the second shell, and 4 in the fourth shell, if this was a typical element. Now this relates to the periodic table because the number on the outer shell, the number of electrons relates to the group, there's this, so group 4, because it's got 4 electrons in the outer shell. And then we've got 1, 2, 3 shells, so we'll say it's period 3. Lesson 4 was the microscope. You need to be able to label this. So first of all, we've got the eyepiece lens. That's what we look through. We've got these 3 other lenses called objective lenses, and we can set these to the right magnification for the job. You would put your sample on the stage, and then you'd switch the light on or use the mirror to reflect light up through that sample so you can see it. Then you'd use your rough focusing knob and your fine focusing knob to make a clearer image. Okay, and this is called the body of the microscope. Lesson 5, we went on to cells. So we got an animal cell and we got a plant cell. Now there's 4 organelles that's familiar with both of these cells. The first one is the nucleus. The nucleus contains the DNA. Then we've got mitochondria, these red things. Mitochondria is the site of respiration. More on that in another lesson. They've also got cytoplasm. That's where chemical reactions take place. And then the last thing that's common is a cell membrane. They both have a cell membrane. Cell membrane allows things to move in and out of the cell. Now if we just focus on the plant cell, the big thing that a plant cell has that an animal cell hasn't is chloroplast, and that's the site of photosynthesis. It's also got a cell wall for strength and support, and it's also got a vacuole for cell sap. Lesson 6 was DNA. So there's not much to know by here, except for the fact that cytosine pairs with guanine and thymine pairs with adenine. So we've got cytosine C, guanine G, thymine T, adenine A. This is found in the nucleus of cells. That was lesson 6. Lesson 7 was on waves. This is your standard wave diagram. We have to be able to label this. So we've got a peak, which is the top of the wave, and we've got a trough, which is the bottom of the wave. The distance from the line to the peak or the line to the trough is called your amplitude. And then one whole cycle of a wave is called your wavelength. So you can see I've gone from the top of the wave all the way down and back up to the top of the wave, and that's where the next wave starts. So if you wanted to know how many waves are here, we've got, if we start here, we've got 1, 2, 2.5 waves. There's two types of waves. We've got a transverse and a longitudinal wave. Now the thing to remember with a transverse wave is that energy is at right angles, 90 degrees. So if the wave direction is going this way, then the energy is going up and down to the direction of the wave. A longitudinal wave, on the other hand, the energy goes in the same direction. So if the wave moves this way, the energy also moves this way. You can see these pockets of compression. That's your energy. That was lesson 7. Lesson 8 then was calculating wave speed. So we need to know that wave speed is represented with a letter V. Wavelength is this letter called lambda, or this symbol called lambda. And frequency is just F. And we've got meters per second for wave speed, meters for wavelength, hertz for frequency. Now frequency is the number of waves that pass a point each second. That's really important. We can use a formula triangle to work out any one of these. So if I'm looking for wavelength, I'm going to put my finger over the wavelength and it's going to be V on top of F, which means V divided by F. If I'm looking for frequency, V is going to be on top of lambda, my wavelength. So it's going to be V divided by wavelength. And if I'm looking for wave speed, I'm just going to times these two together. And that's term 2. Hope you enjoyed.